Thank you so much for that introduction, Jackie. I'm so thrilled to see you all here today and talk to you about a little bit about what I've been up to for the past 6 years, researching kelp physiology in a changing ocean. Um, okay, so if you've ever seen me give a talk before, you've probably seen this slide before. It's one of my favorite ways to open talks because it's one of my favorite art pieces that features bull kelp, which is what I study. So this is a story pole that sits up at Deception Pass State Park, just north of where we are now. And it depicts Kukwalalut, who's also known as the Maiden of Deception Pass. She was a young woman from the Salish Indian nation who made a huge sacrifice for her people by marrying a man of the sea in order to ensure bountiful and continued ocean resources for her people. But as time went on, she became less and less human, and eventually she wasn't able to visit her family on land anymore. But they knew that she was safe and that she was still looking out for them because they could see her hair, which was strands of bull kelp, floating at the water's surface when the tide changed. What I love about this story is I think it so perfectly encapsulates the role that kelp serve in their community. So Kukwalalut herself is this protector and provider figure. And if we are sum up the ecosystem services that kelp provide, it really is this protector and provider figure for the ecosystem. Kelp are what we call a foundation species. They provide food for a lot of our herbivores, like our urchins and our abalone. This in turn feeds these upper trophic levels, like our sunflower sea stars, a lot of our fish and our pinniped species. Most critically, the canopy forming kelp create this three-dimensional habitat stretching from the benthos to the surface that is a really critical area for fish and other organisms to hang out and live. In particular, a lot of our critical fishery species, such as our juvenile salmon and our rockfish, obligatorily use kelp forests as habitat. Kelp forests are often called the rainforests of the sea because of the incredible biodiversity that they host. And they are a very fun place for us to recreate in as humans. And they also, because of this biodiversity, support our upper trophic levels like our otters and our orca. So they're really integral to nearshore marine habitats. And so for this reason, it's concerning that in some areas kelp are declining because we lose all of that biodiversity, we lose all of that critical habitat. Now, if we zoom out and look at this as sort of a global phenomenon, just over a third of kelp beds are declining. So it's not an issue everywhere around the world, but in the areas where kelp are declining, they are declining to a very extreme degree. So for example, after the 2014 to 2015 marine heat wave that hit the Northeast Pacific, Northern California lost 95% of their kelp canopy cover in some areas. And where we are now in Puget Sound, we haven't seen declines that extreme, but in South Puget Sound, we have lost about 2/3 of our canopy cover. And the reasons for these declines are complex, but at least one of the main drivers of these declines are warming ocean temperatures. The thing to remember, though, is that ocean warming is not happening in a vacuum. So anthropogenically derived atmospheric CO2 is also causing ocean acidification. And the important thing to remember about these two stressors is that they reinforce each other, both chemically and biologically. And so because they will so tightly co-occur in future oceans, it's really important to understand their effects in tandem on organismal physiology. And we have a pretty good handle on how ocean warming is impacting kelps because we can go out into the field after marine heat waves, observe that all kelp died. That's pretty clear cause and effect. But with ocean acidification, the story is a little bit more nuanced. And that's because, like other primary producers, kelp are taking up carbon dioxide as part of their photosynthetic process. And so hypothetically, if you're adding more CO2 to the ocean, they're like, oh, great, we can photosynthesize more. This is awesome. This is obviously an oversimplification because the reality is there are a lot of weird things that low pH can do to you at the cellular level that can mess up physical processes. But in general, kind of the consensus in the scientific community is that we expect acidification to either have a positive or neutral impact on kelps. And in this talk, I will be using both PCO2 and pH kind of interchangeably, but as a reminder, so PCO2 is the partial pressure of carbon dioxide in the water, so that's going to increase under acidification. pH is a measure of acidity, so that's going to decrease under acidification. And so when we have these two drivers that are co-occurring, one of them warming, we know is going to be very bad for them. One of them acidification, maybe neutral, maybe positive. When we combine these, really unexpected things can happen, and not many people have studied this. So that was one of the main goals of my thesis work. So roadmap of what we're going to talk about today. First, we're going to think about how climate change impacts the physiology of kelps on a global scale. Then we're going to zoom in and focus on one specific species in a specific region. So looking at how climate change impacts bull kelp in Puget Sound. And then finally, we're going to move kind of beyond the organismal physiology level to think about more like ecosystem-wide issues and talk about carbon contributions of local kelp forests. So to start, we're going to discuss how climate change impacts kelps globally. We're going to discuss this through the lens of a meta-analysis that was recently published in Ecological Monographs. Go check it out. So the literature on kelp in response to climate change has been expanding rapidly. So this graph shows the number of manipulative experiments on true kelps, the number of studies published on this topic through time. And you can see that there's been a massive increase in this type of study conducted over the past decade. And generally, when you have this kind of explosion in the literature, it can be a really fruitful time to take a step back and sort of review what people are finding as a whole, and do a literature review or a meta-analysis. Another thing I wanted to point out on this graph is that our light orange bars are telling us the studies that have been done on both co-occurring acidification and warming, and we can see that there have not been very many studies done on that topic. The issue with sometimes doing direct comparisons between studies is it can be really hard to directly compare different papers that are either studying different physiological endpoints, or they have very different ways of measuring the same thing. So for example, these are all valid ways to measure growth rates in seaweed, but can be hard to directly compare. The beauty of a meta-analysis is it allows you to do those direct comparisons, even if the studies didn't measure the exact same thing in the exact same way. And it does this through this metric called effect size, which is basically the measure of the difference between the data you get under your control conditions here in blue and the data you get under your whatever your treatment condition is, warming, acidification, nutrients, whatever. And if you find really different results under your control and treatment, your effect size is going to be really large. And if you find really similar results under your control and treatment conditions, the effect size is going to be quite small. So we synthesized data from 143 studies around the world. All of these points are showing individual studies, and concerning 42 kelp species. And we sought to answer a couple of questions with this database. First of all, we wanted to see how warming and acidification would affect kelps in general, and then also across different physiological endpoints, different life stages, and different regions. And we did this through running a series of linear mixed effects models, with our effects being duration and the magnitude of warming and acidification, and our random effects being study, species, and experiment. So to first answer our main question, how do warming and acidification affect kelps? This data on the next few slides will be presented as forest plots. And if you aren't familiar with reading a forest plot, we're going to do a quick tutorial. So on our y-axis, we're going to have some kind of categorical division of the data. So in this case, our 3 different stressor scenarios. On our x-axis, we're going to have effect size. And what you're going to want to pay attention to is how these points lie in relation to our 0 line. So these points are going to represent your mean and your 95% confidence interval for the calculated effect size. And so if the error bars do not intersect with your 0 line, that tells us that the calculated effect size is statistically significant from 0. These results have been coded so that a negative effect size is going to indicate a negative effect of the treatment and will be coded in red. And a positive effect size will be coded in blue and will indicate a positive effect of this treatment. Statistically non-significant points will be coded in gray. And I would also like you to pay attention to the size of the data points. That's going to indicate the amount of data that we have behind this data point. And the error bars are going to imply either basically the amount of evidence that you have behind this finding. So, or the degree of agreement between studies. So small error bars are going to imply greater consensus between studies, whereas large error bars are going to imply basically that the studies found a variability in their responses. So with this information, let's interpret this data. We can see that warming has a consistently negative effect on kelps. In contrast, acidification generally has a neutral effect on kelps. And then with these two stressors in tandem, we can see, first of all, we don't have a lot of data here. And the data we do have is really, really variable. So people are finding very different things across different studies. Okay, moving on to thinking about different physiological endpoints. When we break up the data like this, and this graph will be presented in 3 panels. So we'll have warming, acidification, and then multiple stressors. So starting with warming, we can see that warming is bad for kelp across all physiological levels. In contrast, acidification is generally neutral. Sometimes the effects are negative, sometimes the effects are positive. The only statistically significantly negative impact of acidification is on reproduction. And then when we combine these stressors, we can see a very similar pattern happening as with all the data. Is basically there's not a lot of data, and the data we have is very variable, so it doesn't really tell us a whole lot. But what I really want to point out here is this finding, which is that reproduction is the only metric that is negatively affected by both warming and acidification. Okay, so now we're going to break up the data by life stage, but first we need to know what those life stages are. Kelp have kind of a complex life cycle. They have what's known as a biphasic life history or alternation of generations. And so this means that the individuals that we're seeing out in the field, which are called our adult sporophytes, are really only half the story. So when these guys are reproductively mature, they're going to develop these spores patches or sori on their blades, which are bundles of spores. Those spores will be released into the water column, and they are microscopic, and they will germinate, kind of like if you can picture like a seed growing, and they will grow into these gametophytes. These gametophytes are now haploid, in contrast to their parents, which are diploid. And these are microscopic, but they are full adults. So we have generation 1 and generation 2. When the gametophytes are reproductively mature, the males will release sperm to fertilize eggs on the female, which will develop into a zygote and then eventually grow into a juvenile sporophyte. So again, we have generation 1, generation 2, and then generation 3 back here again. Okay, so with this information, now we can split up our data by life stage. And what we can see is, again, that for warming is kind of just negatively impacting all life stages. But there is kind of this interesting pattern where our adult sporophytes, the magnitude of that effect size is maybe a little bit larger for the sporophytes than it is for our microscopic life stages. And then interestingly, with acidification, we see it's non-significant, but we see kind of the opposite trend where our microscopic stages are perhaps more negatively impacted by acidification, and our macroscopic life stages are perhaps more positively impacted by acidification. And then we see the same thing when we combine these drivers. It's just there's a lot of variation between studies. Okay, so moving on to regional trends. If we break up all the data by just general global region, we can see the area that we are in, the temperate northern Pacific, has a lot of data showing that kelp in this region are vulnerable to warming. However, if we zoom in specifically to where we are right now, the Salish Sea, we can see that we actually have no data on the effects of acidification or the effects of these two drivers together. So this is a really important gap in the literature that I sought to fill with my later thesis chapters. Okay, so in conclusion, we saw from this work that warming has a really strong negative impact on kelps. We saw that acidification often does not affect kelps. We saw that the combination of these drivers is pretty understudied, and the results are highly variable. We saw that reproduction was the only metric that was negatively impacted by both warming and acidification. We saw there were maybe some opposite trends happening between our macroscopic and our microscopic life stages, and that the Salish Sea is a really understudied region. So now let's move into the Salish Sea and do some research. So we are here in Seattle, and for part two that we're moving into now, we are going to just be focusing on Puget Sound, and then later in the talk, we'll move up into the Strait a little bit. And before we dive into the research, I wanted to return to that statistic that I showed at the beginning of this talk that showed that, in South Puget Sound, they lost about 2/3 of their canopy. So we're going to zoom into South Puget Sound for a second and look at that on a map. So this map is showing the canopy extent of kelp beds, both historic and current in South Puget Sound. And so our blue lines are going to show us where we historically had kelp and no longer have kelp. And our pink spots are going to show us where we still have kelp. And anecdotally, Andy and I observed that this spot is gone as of last summer. So yeah, we've lost a lot of our kelp in South Puget Sound, which is very distressing. Again, when you think about that contrast between this really biodiverse habitat and then nothing. Clearly, understanding why kelp is declining is very top of mind for the Washington kelp research community. And so I was really interested for some of my thesis chapters in answering how climate change affects, and specifically warming and acidification, affect the physiology of bull kelp, Nereocystis luciana, which is our only canopy-forming species in Puget Sound and the primary canopy-forming species in most of the Salish Sea. And so to look at this, I collected individuals from 2 different kelp beds in Washington state. These are 2 populations that have been shown to be genetically distinct by work done by others. The first is up here on Whidbey Island, so right here kind of at the mouth of Puget Sound and Admiralty Strait. And the second site is here in Seattle in Elliott Bay. And these sites are really different in terms of their environmental conditions and kind of overall vibe. The site up on Whidbey Island is at Fort Casey. It's in a state park. It's very natural, protected. It's in the strait, so it gets really, really strong tidal currents. And in contrast to Elliott Bay, it's the little kelp bed I visited was the one that's like right next to Seattle Aquarium. So it's kind of tucked in between 2 of those big piers downtown. And pretty low current, obviously highly urbanized area. And we had loggers out at both of these sites for 2 years. Here's one of the loggers pictured here. And if we characterize kind of the environmental conditions, Whidbey Island is a little bit colder, a little bit more acidic, again, because it's closer to the open ocean, so that makes sense. And in addition to these differences in environment, I mentioned that these populations are genetically distinct, and they also have very different phenotypes. So our kelp blades at Whidbey Island tend to be really large and thick, and our kelp blades at Elliott Bay tend to be pretty small and thin. And this is likely related to the flow regimes at these sites. Kelp tend to bulk up under strong currents. And so I mentioned that Whidbey Island has really strong currents, and so correspondingly, the blades, the kelp there is very large and thick. And we also see this reflected in terms of overall blade size. So blades at Whidbey Island tend to be about twice the size or longer than blades at Elliott Bay. We can see Emily here as our model for scale in this photo. So we were really interested in understanding how warming and acidification affect bull kelp photosynthesis. So to answer this question, we visited these sites, we collected blades, we brought them back to lab, and we exposed them to a suite of different temperature and pH conditions, with this 10 degrees Celsius pH of 8 being the closest to what they would experience in the field. So that's our control. And our goal here was to construct thermal performance curves. So we weren't going for, super accurate conditions, I guess, for what they would experience in the field or what they would experience in future oceans, because 30 degrees Celsius, that's like tropical conditions. So they're definitely not going to experience anything that hot ever. But we really wanted to push them to their physiological limit to understand where that limit was. And then we also challenged them with 2 different acidification scenarios. So this is sort of a moderate level of acidification, and this is a really extreme level of acidification. And we did very short-term exposures of 1 to 3 hours. We just, the beauty of doing photosynthetic measurements is that they respond very, very rapidly to change. So you can expose them for a few hours and still get really good data. So the first metric I want to talk about here is our maximum photochemical quantum yield. or FV over FM. This is a really common metric used by people that study photosynthesizing organisms to assess health. And basically, it is a measure of photosynthetic efficiency. So we interpret high values of this proportion as meaning that the organism is photosynthesizing efficiently, and low values of this proportion indicate that the organism is not photosynthesizing efficiently. So we interpret low values of this as a sign of stress in the organism. So on these graphs, we're going to have our two sites faceted here. We're going to have our temperature treatments on the x-axis, and we're going to have our different pH treatments in these colors. And what you've probably observed right now is that there's a really strong effect of temperature. As temperature increases, FE over FM declines, indicating that as temperature increases, they're becoming more stressed. There's also this kind of interesting pattern here. If we zoom in on Whidbey Island, we can see that at this really extreme temperature of 30 degrees Celsius, our green dot, which is going to indicate our control pH, is doing a lot worse than our two acidification challenges. So at this temperature, acidification is giving them a little bit of a physiological boost and helping them combat the negative effects of warming, but it's not enough to fully compensate for the effects of warming. Okay, so the next metric I want to talk about is our non-photochemical quenching rate, or NPQ. So this is a metric of photoprotection. So we interpret this as a stress response metric. So it's something we would expect to be upregulated when the organism is stressed and attempting to respond to that stress. And so correspondingly, we see at sort of these temperatures that they are more likely to experience in the real world. So really the 5 to 15 degrees Celsius, NPQ stays pretty low. Then when we get to these really challenging temperatures of 20 to 25 degrees Celsius, NPQ is strongly upregulated. So they are clearly attempting to compensate and respond to the stress. And then once you get to 30 degrees Celsius, they've kind of just given up and they're no longer able to compensate physiologically for the stress. We've learned about our adult sporophytes, but obviously that's only giving us half the story. So now we're going to turn and see how these same climate change variables, warming and acidification, affect our microscopic life stages. And this is a little trickier because obviously we can't go out in the field and observe what's happening to them because they're too tiny. So we kind of have to bring them back into the lab and expose them to these conditions to understand what's going on. Another important thing to know about the bull kelp life cycle in our local waters is that they are an annual species. So that transition between sporophyte and gametophyte happens every single year. In the spring, we see our juvenile sporophytes recruiting and growing until they reach the surface. Once they are mature, they will start to drop these zoospores. And we presume that starting in the fall and going into the winter, our zoospores are germinating and the gametophytes are growing and maturing and reproducing. Obviously, we can't observe that. All we know is starting again in the spring, we see the juvenile sporophytes popping up again. So we revisited these sites the following summer, and we collected these reproductive bundles of spores, or sori. We brought them back to lab. We induced them to release their zoospores, which you basically do by getting them dry, getting them cold, and then putting them back in water. And we grew out our gametophytes in culture in these little petri dishes or well plates for about a month. So each of these little kind of like star looking guys is an individual gametophyte. And so for our control conditions, we used 12 degrees Celsius. And then for our elevated temperature conditions, we used 18 degrees Celsius because that was pretty close to what we were seeing was making the sporophytes very angry. And also is kind of sort of the maximum temperature we're seeing on the benthos here in Puget Sound. And then we chose an acidification challenge of 7.5, so pretty severe acidification scenario. And we measured a lot of things, but the ones I'm going to talk about today are germination rate, growth rate, fecundity, and reproductive success. So we're going to start with germination rate. So this is the proportion of zoospores that successfully make it from here to here, which is a really important developmental stage. First we're going to go through the graph. So on this graph, we have germination rate. On the y-axis, we're going to have our different treatments here on the x-axis, and again, our sites faceted here. And so if we look at the data, we can see that overall, we saw really high rates of germination kind of across all of the different treatments. But if anything, we were actually seeing, and we see some differences between sites too. We see Elliott Bay had kind of overall lower rates of germination. But in general, our climate change scenarios are certainly not hurting germination rate, particularly warming actually seems to be enhancing germination rate. Okay, so I'm not seeing any problems so far. And the story continues when you look at growth rate. So on this graph, we're going to have time now on the x-axis. And our low pH is going to be represented by a dashed line, but otherwise everything should be the same. And then there's a photographic depiction of what we're going to see on the graph here on the left. You'll notice those photographs look quite different from each other. They're the same magnification. And if we look at the graph, we can see that there is a very clear effect of temperature in those red lines, where at elevated temperatures, they are growing way larger and way faster. And we also see the same trend reflected with our juvenile sporophytes. So in these photos, all these little kind of like leaf guys are our juvenile sporophytes. And then this is one giant juvenile sporophyte. So you can see, again, same magnification between photos. They're growing much larger. Okay, when we look at fecundity, so this is number of eggs we're observing per female, we again see something really similar, where we're seeing that warming is either positively impacting or not impacting fecundity. And I think I forgot to mention this in the last slides, but we're really not seeing any strong trends with acidification in general. Okay, so far it seems like warming and acidification are fine for them, maybe even good for them. But the story is about to change as we get to the end of the experiment. So now we're at day 26. They've been exposed to these conditions for almost a month. Now things really start to shift. So again, these photos are at the same magnification and our two different temperature treatments. And again, these photos look quite different. So at our control temperatures, we see that our gametophytes themselves are very, very small, but we see loads of evidence of successful reproduction. So all these little leaf guys are little sporophyte babies that have been produced. And in contrast, we see at high temperature, the gametophytes themselves are quite large, but there's no babies. And we can see this graphically represented here. We're just going to focus on Whidbey Island for this one. And we can see that at our control conditions, they are having really high reproductive success. And then under high temperatures, the reproductive success is very, very low. The other thing I wanted to point out here is that we also see reduced reproductive success under our low pH treatment. So this is a negative effect of acidification, not as strong as the negative effect of warming, but still substantial. So if we're summing up kind of what we saw from this experiment, we saw that warming and acidification was kind of fine through these first developmental stages. But once we got to here, there's some kind of issue. Either we're not sure whether it's pre or post fertilization. So somewhere in this zone, there's an issue. And it's preventing us from reaching this stage. And this is a potentially really critical population bottleneck. Obviously, if your kelp are not reproducing, that's going to be a big problem for a continuance of the population. Okay, so to sum up what we learned from these two experiments, we saw that this 18 to 20 degrees Celsius range of temperatures is a really important physiological threshold. At those temperatures, we saw stress responses activated, we saw photosynthetic efficiency decrease, and we saw reproductive success decrease. In gametophytes, we saw this really striking trade-off between growth and reproduction, where at high temperatures, they basically reallocate all of their energy away from reproduction and towards growth. And these temperatures, and in general, temperatures were really designed to mimic future ocean conditions. But the reality is that we are seeing temperatures this extreme during summer heat waves in Puget Sound. So it does kind of give us a little bit of a window into what's happening right now in extreme circumstances. In contrast to warming, the effects of acidification were subtle and temperature dependent. Sometimes they were positive, mostly they were positive, but sometimes they were a little negative. But certainly it seems like acidification isn't going to be this magic fix for warming. So it's not going to necessarily help them compensate for warming in a meaningful physiological way. we did see some differences in population stress tolerance or timing of different stages. And this is interesting to note because, again, these sites are relatively close together geographically. And so when you're working in an environment like the Salish Sea, which is an inland sea, it's kind of like a giant estuary. And so you have these really strong environmental gradients happening over a really small spatial scale. And so in those instances, it's really important to pay attention to differences that you see between populations and kind of get at whether they're more linked to like acclimation to environmental conditions at those sites, or whether those differences are genetically linked. And then finally, we saw some differences in terms of potentially the thermal maxima of these microscopic stages, which is important because we kind of tend to think of these as a monolith, but the reality is there could be some kind of different things. They could be sort of differentially vulnerable to some of these environmental drivers, and that's really important to parse out. Okay. One sec. Okay, so thus far, we've been really focused on organismal physiology. But for this final part, we're going to kind of step back and move into the rest of the kelp bed and kind of talk about more like ecosystem level, habitat level trends. And so in this part, we are going to explore kind of a debated ecosystem service that kelp forests provide, which is carbon sequestration. Before we dive into this research, it's really important to contextualize it within the broader Washington kelp monitoring and research space, because there's a lot going on in that space right now. And so there's a lot of really cool data to work with at the moment. So I've talked a lot about how kelp is declining in our local waters. And so it should not be shocking that there's a huge concentrated effort going on to document this decline. And one of the main ways we're doing this is through that woman in the back, specifically Danielle, is working to measure, to map kelp canopy extent at a bunch of kelp beds in Washington state. And this is done through a variety of methods, including fixed-wing aircraft, drones, and kayaks, to basically map the linear extent of various kelp beds. And then when this data is compiled through many years, you can see sort of the expansion and contraction of bed area through time. And with this data, you can end up with these really cool time series that are showing whether beds in general, specific beds, are increasing, decreasing, or remaining stable through time. This is a really powerful monitoring tool when you scale it up and do it at a bunch of sites through time. So this is the floating kelp forest indicator for Washington state, which is again run by DNR. And so on this graph, each of these individual points is a kelp forest that is monitored annually. And these points and these sort of general regions are color coded so that cool colors show regions or sites where kelp is remaining steady or increasing. And points or regions that are warm color coded indicate spots where kelp is declining. And so we can sort of start to pick apart these region-wide trends. There's a very clear trend where if you're closer to the ocean, kelp's doing okay. And then if you're further from the ocean, kelp's not doing so great, with some site-by-site exceptions. So obviously we talked a lot about South Puget Sound, and this is obviously an area that's experiencing decline. Also to contextualize, so one of my field sites is here in Seattle, Elliott Bay. And then my other site, Whidbey Island, was just up here. So we can see this is a region that's declining, whereas this is a region that's holding pretty steady. Okay, so in addition to this data set, we also have a lot of data being taken subtidally on species densities. And this is primarily collected by Reef Check, as well as a bunch of partner agencies. And this work has been going on in Washington since 2022. And the mother of Reef Check in Washington is sitting right there. I think right now Reef Check has about 45 volunteer divers and has about 53 sites in the state of Washington, but Jackie can correct me if I'm wrong about that. I've been a volunteer for Reef Check for the past 2 years. It's an awesome agency, and they collect density metrics for both surface and benthic species. And so when you have this information on bed area and you have this information on species density, if you also collect information on individual species biomass, you can end up with what we call a standing crop estimate. So basically, like at one point in time, how much of a single species is there in this area? And there's a lot of reasons why this metric can be useful, but one of those reasons is better understanding how kelp contribute to blue carbon, which is basically carbon sequestration happening in the marine space. So we talked at the beginning of this seminar about how kelp are taking up carbon dioxide via photosynthesis. That carbon is stored in their tissues and then eventually excreted, either in the form of dissolved organic carbon during photosynthesis or as particulate organic carbon as detritus during normal senescence, or if whole plants are ripped out during winter storms. Regardless, that carbon re-enters the system, and we presume that some proportion of it escapes herbivory, escapes microbial degradation, and sinks either into nearshore or deep sea habitats and gets buried in the sediment. And if it is removed from the carbon cycle for at least 100 years, that is considered sequestered. As you can imagine, this is like a lot of coincidences to happen. And so this is certainly a very small proportion of the total kelp-derived carbon meets this fate. But the thing about kelp is that they're so enormously productive that even if like a small percentage of the carbon derived from their tissues ends up here, that's still a really substantial amount. So for example, this is a study that looked at carbon stocks. So how much carbon is there, again, at one point in time, versus carbon flow. So how much carbon flow is entering the system for a bunch of different marine and terrestrial vegetative species. And we can see kelp here at the top is by far the least in terms of the amount of kelp that is there, but contributes an astounding amount of carbon to the detrital food web. So this is, again, showing they're enormously productive. And so this is kind of the reason why people are very interested in this topic. It's very compelling. So if in addition to biomass data, we also collect data on tissue carbon content, this will get us at this metric of carbon standing stock. So at one snapshot in time, how much carbon is being held in kelp tissues in this kelp bed. And this estimate of carbon standing stock is sort of this first step down this long road to eventually getting at the sequestration potential. You also need a bunch of data on primary productivity and on that proportion of dissolved and particulate organic carbon that actually meets that sequestration fate. But this is sort of that first step down that road. And critically, most of the time when people do these carbon standing stock estimates, they kind of focus on whatever the main species is in that region. So normally that's whatever your canopy forming species is. But the beauty of working in the Salish Sea is that we have 22 native species of kelp here. And only 2 of them are canopy forming. So our giant kelp and our bull kelp, everyone else is below the surface. So there's a lot happening underneath the surface that's really important to quantify. Another quirk about working in this region is that in Puget Sound specifically, we have a lot of kelp forests that look very different from the kelp forests that I've been showing thus far, although beautiful photos. There's also a lot of kelp forests that look like this, where there's no canopy to be seen. There's just a lot of understory, in this case, a lot of sugar kelp. And so again, then understanding the role that these understory taxa play in ecosystem dynamics is really important. So for this chapter, I was interested in, first of all, calculating how much carbon is in a Salish Sea kelp bed, how much variation exists between beds, and then how much carbon do individual species contribute to that full carbon standing stock of that bed. So we did this work at 4 different sites in the Salish Sea. And our first step was to establish allometric scaling relationships between morphology and biomass to negate the necessity of doing destructive sampling in the future. I know a lot of people in this room are fish biologists, and there's really well-established relationships between length and weight. And as we've talked about in this presentation, kelp are so phenotypically plastic that we do not have those estimates for the Salish Sea. So that was a critical first step. And then our second step was to quantify those standing stock metrics and really see whether there was consistency between sites, because obviously we can't do this at every single kelp bed in the Salish Sea. So it would be great if we could extrapolate between sites. I'll spoil this and say that there was not consistency between sites. Okay, so I picked these sites to be as different as humanly possible in terms of their geographic location, the environmental variables that are at these sites, so the physical conditions, what the benthos looks like, all of that. And so we ended up with two sites that were pretty, they kind of just had our three like heavy hitters that we see everywhere in the Salish Sea are our bull kelp, and then our two understory kelps are sugar and our five-rib kelp. And then our other two sites, Point Caution and North Beach, which were closer to the ocean, were much more species rich, but sort of had different like uh, species that were most abundant at these sites. And we can see this reflected in our species density data. So here I have our sites grouped from south to north. And the main thing I want to show you here is that each of these sites looks pretty different from each other in terms of which taxa are the most abundant. There are a couple of like surefire ones like sugar kelp. You're always going to see a lot of sugar kelp. That's in that pink bar. But then the other sites, there's kind of different kelp that are sort of really abundant at those sites. The other thing I want to point out is that bull kelp, which is in this purple bar, is never the most common thing that we're measuring underwater. Okay, so to get these estimates of biomass and morphology, we visited each of these sites on scuba, mostly me and that girl right there, Andy. And we measured 15 different individuals for morphological parameters. And then we collected 5 of those individuals to bring back to lab and get biomass estimates. As a reminder, we saw huge differences in morphology in bull kelp in prior projects. And so it should be unsurprising that this also scaled up to this project. So if we look at the average weight of one kelp plant of bull kelp at all of these sites, we can see there's massive differences in average size. And in fact, our two sites that are closest to the ocean, North Beach and Point Caution, have way larger individuals than our northernmost and southernmost sites. And we see this tracking also for our sugar kelp and our five-rib kelp. Now, the interesting thing here is that we don't see consistency in terms of sites in the sense there's not one site where everyone's bigger and one site where everyone's smaller. So it seems like the individual environmental characteristics at these sites are sort of positively or negatively driving morphology in a species-specific manner, which is really interesting. But it also means that it's going to be really hard to extrapolate between sites. And so then developing those allometric scaling relationships becomes even more important. So I was very lucky to use this data set that was collected by primarily by the Washington Department of Natural Resources, where they collected information on a bunch of different morphological parameters of bull kelp, and I was able to relate these to biomass. So these are all the metrics we tested out, and out of these three, so the maximum sub-bowl diameter, which is this difference here, the wet weight of blades, and the maximum blade length, when combined, created the best performing model. However, if you're designing a monitoring program, this is a pretty big ask to ask people to go out and measure all of these. So in a realistic monitoring scenario, you'd only be able to measure one of these, and the easiest to measure and the best performing metric is this maximum sub-bowl diameter. And the cool thing about this finding is this also agrees with what folks in British Columbia and Alaska and some other areas in the West Coast have found. So we have nice agreement between regions that this is a really good estimator of biomass. For our understory taxa, they're kind of, a lot of them are kind of oval shaped. And so unsurprisingly, basically just getting maximum length and maximum width gives a pretty good estimate of biomass. Critically, you need both of those though, not just one. Okay, so on to the main event, which is our actual carbon standing crop estimates. On these graphs, we again have our sites arranged south to north and our species on the bottom, and then our carbon content standardized per square meter on our y-axis. And I'm gonna have these reveal one by one, starting with actually the one over here, which is the only site that performed how I hypothesized they would all perform, which is Cherry Point. And if we look at the carbon content at this site broken up by species, we can see that bull kelp is the main contributor to carbon standing stocks out of all the kelp at these sites. But these really numerically abundant understory taxa like sugar kelp are also contributing a fair amount to carbon stocks. That was not true at any of the other sites. So at Point Caution and Lincoln Park, the vast majority of carbon being held in kelp tissues at this site is being held in sugar kelp tissues. And in comparison, bull kelp isn't doing a whole lot. And then it's even crazier at North Beach, which has a really substantial woody kelp or pterogophora understory. And here, the vast majority of kelp carbon is being held in pterogophora tissues. Okay, So for to sum up what we learned in this part, we saw that sites varied a lot in terms of their species density and composition, which again highlights the critical nature of performing routine subtidal monitoring. We saw that the size of a single kelp plant varied a lot by location, but luckily we were able to develop these allometric relationships that remain fairly consistent between sites. And the beauty of all these metrics is they are measurable in situ, so they could be adapted by existing monitoring efforts to include in those efforts and start to obtain this more widespread information on biomass. And then finally, the big thing we learned is that understory kelps are really significant contributors to carbon stocks in this region. Okay, so to sum up what we've learned over the last 6 years, going from more of a global to a local, and then moving from organism to ecosystem sphere. In part one, we saw that globally, kelps show a lot of vulnerability to warming, but are pretty resilient to acidification. We identified some critical knowledge gaps, which was that the Salish Sea is pretty understudied and the effects of multiple stressors are understudied. We also saw that reproduction was a pretty vulnerable endpoint. Moving to the local scale, we saw that bull kelp in Puget Sound are vulnerable to warming and pretty resilient to acidification. This allowed us to fill in that critical regional data gap on multiple stressors. And we also saw that reproduction was vulnerable to both warming and acidification, which again matches what we saw in terms of global trends. Moving from the organism to ecosystem level, in part three, we talked about how kelp forests show a lot of regional variability in terms of their morphology, their species composition, their species density, and their overall carbon standing stocks. And we really highlighted that understory kelps are really critical contributors to carbon cycling, but not only that, but also nutrient cycling, food web dynamics. They're so abundant and potentially a little understudied. With that, we've reached the end of my talk. So I would just like to say thank you so much to my committee, particularly like my committee chair, Jackie, who's been on this journey with me for the past 6 years. And special thank you to my committee for all the ups and downs we've been over the last few months with all the uncertainties, but we made it here today. Just the biggest thank you to all my funders and collaborators, particularly the Washington Department of Natural Resources that have funded the majority of my research, and I'm so grateful for them, as well as the broader Washington kelp community that has just been invaluable in being able to complete this work. My broader UW support team, which includes the diving and boating safety team, John, our facilities director, that I genuinely could not have finished this degree without his help, and all of the volunteers and undergrads that have helped me with my research. Again, I would not have been able to do it without you guys, and I'm just so proud of all of you and where you've gone in your journeys. The Padilla-Gamiño lab is very special to me, and I've made some lifelong friends of this group. I'm very, very grateful to them, as well as the broader SAFS grad community. I think everyone in their defense talks thanks the SAFS grad community, and it's because it's such a special group of people. Again, like lifelong friends, could not have done it without you guys. And my general friend group, just so grateful to you all for your unyielding support. I'd also like to thank all of my friends' pets. As a lot of you guys know, my side hustle in grad school was being a pet sitter. But my secret was, and for a lot of times for people that are in this room or on Zoom, but you guys were paying me to watch your pets, but I was getting free therapy out of it. So I appreciate it. And thank you to my family, particularly my mom and my aunt that are here today, and my lovely sister. And my friends that are basically my family at this point. And you guys are great. And okay, the next slide I'm going to try to get through without losing it. So I would also like to thank my dad, Craig. This presentation is dedicated to him. As a lot of you guys know, we lost him about 3 years ago, very suddenly. So that was right at the midpoint of my degree. And, you know, during the first half of this degree, I imagined a lot of things going wrong today, but never in my worst nightmares did I imagine him not being here. It was not something that ever occurred to me. He was my biggest supporter and champion throughout the first 3 decades of my life. He instilled a really deep love in nature in me from a young age. He and I were always the ones that would just like get into any body of water, whether it was a pool or a lake or whatever that we could find. He loved animals, like probably way more than humans. Like these pictures are like exactly how he liked to be at all times, just like surrounded by dogs. Um, and since he's not here, I wanted to kind of say a few things that I think he would be doing if he was here. Um, he was the total stage mom. So um, I think what he would be doing is he'd be sitting right where Luke is right now on the edge and be getting up periodically to like kneel and like take like candid photos with like a really aggressive, like way too fancy camera. And it would be like really disruptive and all of the photos would turn out really unflattering. And then, you know, at the end, he was a very active listener. So he'd ask a lot of questions. Maybe they would not be relevant to the talk, but that's okay. And then during the closed door session, anyone that was hanging around out there, he'd corner each and every one of you to like deeply interrogate you about your research and what you did. No STEM background. He just like loved to learn and loved to meet new people. And so one of the things that makes me saddest about not having him here today is that you guys all didn't get to meet him and experience the light and joy that he exuded into the world. So I'm extremely cognizant of the fact that I would not have made it here today without his, you know, almost 30 years of his steadfast support and love. So I wanted to acknowledge that and say thanks, Dad. Okay, and with that, I'd like to thank everyone so much for coming. And if we have time for questions, I'll happily answer any.
This was amazing, and you are so incredible, and I am so proud of you. This is beautiful. I remember when you were 8, when you wanted to be a marine biologist, and I said I did too because you did, but I didn't want to. And you did, and you became a marine biologist, and that's incredible.
Mhm. Is it because of Yeah, that's a great question. So yeah, CR situation is interesting. So if we zoom back, like way back several centuries ago, obviously the fur trade really decimated otter populations up and down the west coast of North America. We are slowly increasing those numbers, but it's been a real challenge, and so there are still a lot of areas where sea otters are completely. Yeah, that's a great question. So yeah, CR situation is interesting. So if we zoom back, like way back several centuries ago, obviously the fur trade really decimated otter populations up and down the west coast of North America. We are slowly increasing those numbers, but it's been a real challenge, and so there are still a lot of areas where sea otters are completely gone and still missing. And in fact, I think historically we did have sea otters in the Salish Sea, but we basically don't see them except on the outer coast. There's been like rumors and sightings of like one or another in the San Juans, but they're pretty much non-existent. And the reason why that's so critical is because sea otters are what we call keystone predators. So they exert this really important top-down control over the kelp forest ecosystems because they eat the things that eat kelp. So sea otters eat like an insane amount of urchins, for example, and urchins are really voracious grazers of kelp. And so one of the main reasons why I mentioned Northern California lost a lot of their kelp forests, I said it was due to warming, but it's actually a little more complicated than that, which is basically in conjunction with this marine heat wave, there was an explosion in the population of sea urchins. And so they completely overgrazed the kelp forests so that there was just like barrens left behind, what we call urchin barrens. And one of the reasons why there was this explosion in population of urchins is because we didn't have those sea otters sort of at the top of the food web to control the urchin population. Andy.
Do you have a field site that is your favorite? Any reason?
Oh, that's a good question. I'm gonna say Lincoln Park, because I think we've logged the most hours there on all of our field sites. And it's like very predictable, very nice. It never does weird current things. Yeah, yeah. Ariel?
Thinking about acidification stuff really interesting.
Yeah.
There's a lot of freshwater input. The sound area.
Yep.
Do you have any thoughts about that? Maybe that be part of the resiliency against acidification, or maybe just in a different way, like that might be changing, and that swing of that input may help will help resilience. Have we thought about that versus like coastal areas?
Yeah, that's a really interesting point, and definitely something I thought about. I didn't show these graphics in this presentation, but one of the other like sort of nuanced differences we saw in the photosynthetic response of those two different sites was that the Elliott Bay population seemed like it was maybe a little more resilient to those increased temperatures under certain like photosynthetic parameters. And I kind of had this hypothesis going into this experiment that like, the overall like physical health of the population at Elliott Bay is probably a little bit lower because they're exposed to less ideal conditions. But like you said, they're really used to quickly acclimatizing or acclimating to these fluctuations, whether it's temperature, salinity in particular, pH, all of that, because they're in such a like dynamic environment in Central Puget Sound. And I think that the fact, and we kind of did see that reflected in some of the data, like their overall health metrics were a little bit lower, but they also seemed like a little more resilient to responding to those changes. So I feel like that's where it kind of was reflecting in their physiology. Yeah. Bishi?
I had a question related to kind of the great talk. Thanks. Kind of species composition at your different sites, and how they change over time.
Yeah.
Like whether I think about a rainforest, a big tree falls, and have all the faster growing trees coming in first, and then outcompeted by the big slow growing tree. Is that something that happens in kelp with the different kinds of kelp, and how would that affect?
Absolutely. So yeah, I mean, because these kelp forests are sort of working on an annual timescale, like the understory taxa don't die off completely like the canopy does, but really, I mean, like if you visit these sites during the winter, it's pretty barren, and then you visit during the summer and it's a whole rainforest, right? So we do have this like really strong annual turnover, and there are definitely some differences that can happen on the annual scale. That was beyond the scope of this project, but like is definitely really, really critical to investigate. And there is a lot of competition that happens in terms of like, in terms of space and light, just like in a terrestrial forest. And there is a lot of, kind of the phenomenon where whoever gets there first wins. So there's definitely a lot of that going on. Taylor?
In your second study, you identified that there was a bottleneck happening somewhere between the gametophytes becoming sexually mature and actually successfully reproducing at high temperatures. Do you have any hypotheses about like where exactly that bottleneck lies, or what mechanism is limiting the system?
Yeah, that's an excellent question. So yeah, we're not sure whether it's happening pre or post fertilization, because I mean, the hinge point is that fecundity metric and the number of eggs we're observing per female, and we didn't differentiate between fertilized or unfertilized eggs, which would get at that. I think it's likely that there's like, if I was to think about sort of the physiological pathway, I think it's likely that sperm could be negatively impacted by these stressors because they're just little guys. They're very vulnerable. They don't have a lot of like cellular protection. And I also think it's extremely likely that there's actually no like problem with like there's nothing that's like, I guess, like directly inhibited or not something that's like by these high temperatures. It's more like, as we know, that sporophyte stage, the production of that sporophyte stage is really triggered by some combination of environmental parameters. We think temperature and light primarily, but also maybe nutrients. We don't really have a good handle on it. And so we presume that if they don't receive that exact trigger being like, okay, it's time to make babies because your babies will be happy, they just will not make babies. And so I think, to get out all these things, you do a couple of experiments. First, you do an experiment that's where you're really, looking at their ability to produce sporophytes across an entire environmental gradient to really nail down what that trigger is to produce sporophytes. I think also really critically, you go back in and you really differentiate between those fertilized and unfertilized eggs. And then I'm right now I'm working on a proposal for I kind of where I want to go next, I guess, is think about how the conditions that the zoospores experience while they're in the sori at the surface, which is a patch of water that is really warm, whether that has an impact on later gametophyte health and ability to reproduce. Yeah, that was a long answer to your question. Tom?
Taylor, Meg asked that same question, so way to go, Taylor. Great
Yeah.
answer.
No. We don't. We can positively identify. So when we are first able to like see bull kelp with our eyes, the little recruits, they kind of just look like little green guys about this big. And spoiler alert, all of the different kelp species recruits also look like that. So we determined through a very scientific analysis this past year that they get about this tall, and then you can tell. What do you think? Like this tall?
Oh.
Okay, this tall. You can tell how big they are, what species it is, and you can identify bull kelp. But until then, not a clue what's happening with them or what the timing is. Okay, Eileen?
The gametophytes, the little star guys,
Yeah.
if you're looking at microscopically, are they like negatively buoyant? Like, do they sit just at the kelp forest, or do they like bop around really far in the water column? And what are the implications of that for like kelp forests that have died out in one area coming back?
Yeah, such a good question. So they're like sticky, so they'll like stick to the bottom. But critically, you can like each of those cells, you can rip apart a gametophyte, you can chop it up into little chunks, and then each of those will like grow into its own organism. So for example, in kelp culturing facility, like they have at PSRF, one of the main ways that they create these cell lines of clones is they'll literally just grow the gametophytes to be really big, and then they'll stick them in a blender, chop them up, and then you can just make like a clone army. And so when the zoospores settle onto the benthos, they stay there. But we presume that there's also some level of asexual reproduction happening where they are sort of like parts of the gametophytes are breaking off and cloning themselves. But that's all like deeply uncertain and hard to quantify or observe in situ. Callum?
Yeah, excellent job. I was thinking about the phenotypic plasticity you had between the two sites for the bull kelp. And forgive me if you mentioned something like this, but did you see a similar kind of plasticity for those other understory species as well? Was there some more generic, like, the ones in the higher flow, which are bulkier, and that might have something to do with also their carbon sequestration?
That's a great question. So yeah, if we look at our differences in morphology we saw between sites, we can see that there's not a lot of even amongst, so it might make sense that so you might have different selective pressures on our canopy versus our understory species. But even if we compare our two understory species, you can see really different things happening. And this could be an issue of succession, like Leachy was getting at, like, someone got there first and so they did better. But I would say in general, kind of the disparity that we're seeing here is telling us that things are a little more like, yeah, it's hard to pick out like what exactly is driving these differences in morphology. I think we would need a lot more data from a lot more sites to start really get at that. Yeah, great question. Jackie?
Sticking with some of the biomass things, you found that there was a correlation between cell wall size and biomass and
Stipitate. Yeah, great question. So only one of my sites, North Beach, had stipitate kelp at it. And I didn't mention it because we tried to get metrics for Pterogophora, woody kelp, and we really couldn't come up with good metrics. We tried, morphology parameters that they've used in California. I couldn't get any good estimates that way. But I was working with a small sample size, so I'd love to repeat it with a larger sample size. And I honestly think trying some of the things that worked for bull kelp, like looking at stipe width and stuff like that, would be really promising. So yeah, it remains to be seen.
So it's time to move to the next stage of the doctoral exam.